Hello class. So now I welcome you to another lecture, and in today's class we will see that how come or how is the general mechanisms of the enzyme activity and what models they were actually presented by the scientists to justify that how the enzymes they are working in our body. Now, if we don't go further, but if I just recall some of the things from the previous class, so I have told you that in the enzymes we have this space, or we have got this location where the substrate is gonna bind, and this would be entitled as, or this would be termed as an active site. And that reactant particle or the reactant molecule on which the enzymes are gonna work or the enzymes they would be catalyzing. So we were referring to this reactant particle to be referred to as or to be called as the substrate particle or the substrate molecule. Like every enzyme would work on a reactant, on its specific reactant, and that specific reactant is entitled as a substrate. Now here I will quickly take you that how come the general mechanism of the enzymes actually look like. So you see here I have the enzyme molecule, here I have got the substrate. Now against the active site of the enzyme we have two substrates. Like here I have one substrate and here I have the second substrate. If we were to be looking on toward their shape, so you see that this substrate has the shape which is complementary to the enzyme's active site. Like it can bind to the active site. But this substrate that I have got over here, so the shape of the active site is different from this. It is not complementary to this substrate. So normally in our body, these enzymes and the substrate molecules that we have, so they move randomly. And when they are moving randomly, so there is a very greater chance that these substrate and enzyme molecules they would collide with each other. But when they are colliding, so that substrate whose shape is complementary to the enzyme's active site that I have got over here, so they would bind with one another. And when they would bind with one another, so as a result of that, we would make a complex out of it, and it would be entitled as an enzyme substrate complex, we say. So at this point, this is an enzyme substrate complex. And further, this enzyme substrate complex would be broken down, and we will have our enzyme molecule unchanged. This would remain intact. Like the enzyme has contributed the role in the chemical reaction, but at the end, we have got it as it is. So this is an important character or the feature of the enzyme. And this substrate that we bonded into the enzyme's active site. So here you see that this has got into its respective product. Now, here comes the hypothesis that the scientists they basically presented that how the enzymes they would be working in our body. So when we talk about the word hypothesis, we say this is a simple assumption or this is the simple idea which is entirely based on our prior knowledge. So, two of the hypotheses were basically presented by the scientists in order to justify that how the enzymes that we have in our body they are going to be catalyzing their respective substrate. So, one of the model was termed as the lock and key hypothesis, and the second model that we have got with us, so it was termed as an induced fit model. Now, if I were to take you toward the lock and key hypothesis, so you may have seen in your daily life that we have got the locks in our homes and we have got the keys in our homes. And this side of the lock has to be complementary to the shape of the key. And in that way, the key would fix here and it is gonna open up, or this would open up this lock. So likewise in our body, you have to just feel is that the enzymes that we have got, so they are acting as a lock. So I'm saying the enzyme they act as a lock, and the substrate particles that we have with us, so they would act as a key. So that substrate whose shape is complementary to the enzyme's active site, so this would bind with the enzyme's active site, and as a result of that, we would make an enzyme substrate complex out of it. And this enzyme substrate complex would be further broken down into the enzyme. Like I could not say further broken down, but rather than the substrate that we have here, which is acting as a key. So this would be broken down into the respective products, and the enzyme has come intact. So if you are required to be remembering the explanation for this, so how you would be describing it or explaining it to your examiner, you would say that as in the daily life, when there comes a specific key and it opens up a specific lock. So likewise for the enzymatic reaction, any of the substrates that we have got for the enzymatic catalysis, so this would act as a key. An enzyme, on the other hand, it would act as a lock. And that substrate, among many, whose shape would be complementary to the enzyme's active site, so it would bind with it, and as a result of that, we would end up making an enzyme substrate complex out of it. And that enzyme substrate complex further would revert out into giving our enzyme intact, and we would end up making the product out of it. And you see that in this lock and key hypothesis, on the arrival of the substrate, the enzyme is not changing its shape of the active site. So as per this model, the enzyme molecule is rigid. This is not flexible. Now, further, the scientists they came up with another kind of a mechanism, and this mechanism it was termed as induced fit model. And what the induced fit model basically presented that the enzyme that we have over here, so if you were to look onto it, the shape of its active site, so this is not completely complementary to the substrate molecule, isn't it? But as a substrate would come and it would collide within the enzyme's active site, so this enzyme is going to slightly mold the shape of its active site. It's slightly going to change the shape of its active site so that it can tightly bind the substrate molecule into its active site. So how we say is that then the scientists they presented is that the shape of the enzyme active site in some scenarios this is not perfectly complementary to the substrate, but as the substrate would approach the enzyme molecule, so the enzyme would slightly alter the shape of its active site. And when it would slightly alter the shape of its active site, so the substrate would tightly bind with the enzyme's active site to be making an enzyme substrate complex, and we will have our enzyme at the end, and the substrate would be converted into product. So if I were to conclude these two models, that what things we need to know is that as per the lock and key hypothesis, the enzyme is a rigid molecule. And as per the induced fit model, the enzyme is a flexible molecule. And as per this model, for every separate substrate, we require a specific enzyme. But as for the induced fit model is concerned, then that means that if we have got a large amount of the substrate and we would need very fewer enzymes against it because enzymes they are in the position to slightly change the shape of its active site. And as of the recent research, the induced fit model is more widely accepted because in our daily life we eat a lot of stuff but against that, a lot of variety that we are consuming in our daily life, the variety of the enzymes is very less. And how this is possible? Because enzyme molecules, they are flexible. They can slightly change the shape of an active site. So against multiple substrate, one enzyme or fewer enzymes would be enough to do the work. So I hope you would be clear with the general mechanisms of the enzyme's activity, the two hypotheses in terms of the lock and key hypothesis and the induced fit model. So see you in the next class for the factors that what factors we have that would be changing the activity of the enzymes. So see you till now. Take care.